Chapter 14, Segment 2: The Genetic Basis of Olfactory Receptors. Molecular biologists Linda Buck and Richard Axel showed that the mammalian genome contains between 1,000 and 2,000 different odorant receptor genes, each which codes for a single type of odorant receptor. The odorant receptor genome is the largest gene family known in mammals. Pseudogenes are the genes that are present on the chromosomes, but the proteins coded for by the genes never get produced. In humans, the odorant receptor genome contains about 1,000 genes, and the proportion of pseudogenes is between 60% and 70%. High proportion of odorant receptor pseudogene in humans is the result of an evolutionary trade-off between vision and olfaction. Old World primate species have about 30%, whereas New World species have about 18%. Howler monkeys have something in common with the Old World primates that the New World monkeys do not: trichromatic color vision. The more copies of a specific receptor you have, the more sensitive you will be to certain odorants. Also, whether you have the pseudogene or a functional gene for a given receptor also alters odor perception. An example is people who hate the aroma of cilantro are missing the gene for detecting the herbal floral component of this aroma, and therefore only detect the soapy note. Strong scents in general tend to be perceived as less pleasant than the same scents at a lower sensitivity. Having few receptors of a given type can lead us to perceive a scent weakly, and if we like an aroma, we may expose ourselves to more of it to get the same punch as someone with more odorant receptors that are sensitive to that odorant. When it comes to food, the more intense our perception of the retronasal aroma of the food, the less we tend to consume of it. Example: if our odorant receptors expression for the odorant that contributes to the banana aroma in banana cream pie is weak, we may eat more pie than someone who can perceive the banana aroma strongly. Our genetic makeup cannot predetermine our response to any given odorant or food. The feel of scent. Our experience of odors often has a feel to it, as well as a smell. Most odorants stimulate the somatosensory system to some degree through polymotor nociceptors inside the nose. The trigeminal nerve is the fifth cranial nerve which transmits information about the feel of an odorant, as well as pain, irritation sensations. Why we tear up when we chop with onions and why we sneeze when we sniff pepper is because of this. High levels of this stimulation can produce a severe burning sensation and has been linked to the facial head pain felt in migration headaches. From chemicals to smell. The shape pattern theory is the current dominant biochemicals come to be perceived as specific odors. Shape pattern theories contends that different scents as a function of the fit between odor and shape to odorant receptors shape activate different arrays of olfactory receptors in the olfactory epithelia. These various arrays produce specific firming patterns of neurons in the olfactory bulb, which then determine the particular scent we perceive. The most recent molecular research suggests that scents are detected by means of a combinatorial code, where one odorant may bind to several different receptors and one receptor may bind several different odorants to varying degrees. This means that different scents activate different arrays of olfactory receptors in the olfactory epithelia, producing a glomerular activity in the olfactory bulb. The specific pattern of the glomerular activity in one olfactory bulb then determines the particular scent we perceive. Vibration theory is an alternative to shape pattern theory for describing how olfaction works. Vibration theory proposes that every odorant has a different vibration frequency, and that molecules that produce the same vibrational frequencies will smell the same. This was championed most recently by Luca Turnin. Specific anosmia is the inability to smell one specific compound amid otherwise normal smell perception. Specific anosmia are due to faulty odorant receptors interaction or the lack of specific odorant receptors not odorant vibrations. Most specific anosmia are to stereomus compounds, and the condition appears genetic. Stereoisomers can exist in different structural forms in which the spatial arrangements of the atoms are a mirror image rotation of one another, like a right and left hand. At the same time, all molecules vibrate and all receptors that are made of atoms that vibrate. The importance of pattern. Each type of olfactory receptor responds to the structure of certain molecules only. However, a molecule may have features that stimulate several different receptors. As with color detection, we detect odors by the pattern of activity across various different receptors. The intensity of an odor also changes which receptors will be activated, which is why weak and strong exists. Weak and strong concentrations of an odorant do not smell the same. An odorant that activates several receptors will also stimulate them in a specific temporal sequence and speed. Different odor perceptions can be due to different odorant receptors firing patterns, or to firing patterns of the same receptors at a different rate and or in a different sequence. Pattern perception mechanism is that if two odorants, one molecularly simple and other complex, activate the same receptors in the same way, we ended up smelling the same thing. Is odor perception synthetic or analytical? How do we process the components in an odorant mixture? Well, there's analysis and synthesis. Auditory mixtures provide the classical examples of analysis, whereas color mixtures provide the classical example of synthesis. Binaural rivalry is the competition between the two nostrils for color perception. When a different scent is presented to each nostril simultaneously, we perceive each scent to be alternating back and forth with the other and not a blend of the two scents. Olfactory white is when at least 30 odorants of equal intensity that span olfactory, psychochemical, and psychological space are mixed, produces a resultant odor percept that is the same as every other mixture in 30 odorants, meeting the span and equivalent intensity criteria. This is equivalent to white noise. The power of sniffing increases the ability to detect odorants. It produces greater activation in some